So welcome everybody. First, I would like to thank the organizers for inviting me and all of you for coming. As Adam said, my name is Tomasz Górkiewicz and I work in the laboratory of neurobiology of emotions. And today I'm going to tell you about social buffering during fear memory extinction. Social buffering was discovered in humans and then studied in many different social species like non-human primates, monkeys, guinea pigs, and rats, and many others. In this phenomenon, the presence of the partner diminishes the stress response of a distressed individual and allows faster and better recovery from stress. As we know, fear can be both beneficial from the biological point of view, but also detrimental if causes phobias or PTSD. To treat this, exposure-based therapies are used for humans, but the biggest problem of such therapies is that their results are relatively short-lasting. Very often we have renewal of previously extinguished fear after a relatively short period of time. One possible solution for that would be to use group therapies instead of individual approaches, but it still needs to be confirmed. So fear extinction is a well-established animal model of behavioral therapies for humans. And the aim of our study was to investigate the role of social buffering in fear memory extinction. We wanted to check if social buffering can improve fear extinction. For this purpose, we designed and developed the behavioral paradigm for studying social buffering during fear memory extinction. Here we have a scheme of entire experiment. Simply, the rats were kept together for at least 2 weeks to establish and stabilized social bond between them. Next, they were habituated for subsequent 5 days to the experimenter, to the transportation, separation, and to the experimental room. Experimental cage, as you can see here, was divided by wire mesh into 2 separate partitions. It allows limited physical contact for rats, and rats can easily see, smell, and hear each other. On the first day of the experiment, both rats were subjected to fear conditioning to the tone in context A separately. What's important. They received 5 repetitions of the tone co-terminated with a foot shock. During the next 3 days, 1 rat from each pair was extinguished by receiving 30 repetitions of CSs without foot shock in context B, different from the context A. The second rat, the subject, was not extinguished, but only exposed to experimental context, to experimental cage for the same amount of time. On the last day, we did extinction test. This unextinguished subject were subjected to fear extinction together with partner or alone. And here we have results. We had 2 experimental groups. In one group, unextinguished subjects were tested together with partners. In the second group, they were tested alone. First, we calculated the level of freezing at the end of conditioning session to observe if in both groups rats acquire high level of freezing. And indeed, we can observe here that in both rats we have high level of fear. Next, we look at the freezing during the extinction test. We calculated both average freezing after CS and freezing after subsequent CSs during the session. And as you can see here, the rats tested alone did a lot of freezing behavior, but rat tested alone display much less freezing behavior. It means that the presence of the partner buffers fear's response of unextinguished subject. We also did more detailed behavioral analysis, and rats tested with partner did much less prosocial behavior, much less merge sniffing, and much more rearing, while rats tested alone spent much more time motionless. And we also recorded USV vocalization and use it as an additional measure of emotional state of the animals. And we calculate the number both 22 and 50 kilohertz calls. And as you can see here, rats tested together emitted much less 22 kilohertz calls than the rat tested alone, but much more 50 kilohertz than the rat tested alone. In previous experiment, we used two separate physical contexts. Context A for conditioning and context B for extinction. Is standard procedure for extinction animals. But in this case, we have additional context, social context, the presence of the partner. And the rats were conditioned separately and tested together, so the social context was changed. And we know from literature that the extinction is highly context dependent. So the question was whether the fear inhibition effect we observed previously was caused by a social buffering or just by changing the social context. To ask this question, in this variant of our paradigm, the rats were subjected to fear conditioning together simultaneously and then tested alone and compared to the previous experiments where they're conditioned separately. And as you can see here, we observed some increase of level of freezing, but this effect was not very robust. It was too small to explain the effect we observed previously. Rats conditioned together also did less prosocial behavior, but the pattern of ultrasonic vocalization was pretty much the same. In the next experiment, during the test of unextinguished subject, the partner was anesthetized and placed in the middle of his experimental chamber. And in this case, we got very similar results to results during solitary test I showed you in the previous experiment. So there was no social buffering at all. Rats tested with anesthetized partner did almost no prosocial behavior and spent much more time motionless. Also, the pattern of ultrasonic vocalization was very similar to pattern during solitary extinction. The rats emitted almost no 50 kilohertz, but emitted more 22 kilohertz than rat tested with freely moving partner. In the next variant, we wanted to check if visual cues can mediate social buffering, if are important for this effect. And to eliminate them, we use untransparent partition instead of wire mesh as previous. But the subject could still see and smell the partner. And in this case, the magnitude of social buffering was smaller because the level of freezing of unextinguished subject was higher than in group where the rats were separated by wire mesh. But we saw no differences in other behaviors, and also the number of 22 and 50 kilohertz was the same in both groups. Next, we look at the emotional status, because in previous experiments, the partner was extinguished, but the subject did not. So the partner had lower level of freezing than the subject. Here, both rats were not extinguished. So both rats had relatively high, similar level of fear. In this case, we observed some increase of freezing level, but this effect was still not very robust. And we saw no differences in other behaviors. And also the pattern of ultrasonic vocalization was similar. Next, we used as a partner naive rats. So the partner was not subjected to fear conditioning. He was only exposed to experimental cage, habituated to that, but without conditioning. In this case, we observed the same level of freezing, so the same level of social buffering. The naive partner seemed to be as effective as social buffer as previously experienced one. The only difference we saw in prosocial behavior. The unextinguished rats tested with naive partner did less prosocial behaviors. It seems that naive partner is less attractive to interact with. The number of vocalization at 22 and 50 kilohertz frequency was the same in both groups. Two last factors we checked were familiarity and strain sameness. We wanted to see if those factors can influence the magnitude of social buffering in our paradigm. So in this case, we use as a partner naive rats from the same strain, Longevans, the same sex, the same age, but totally unknown to the subject, or from different strain. In this case, it was Longevans rats. Here we have results. In both groups, we observed higher level of fear than in subject tested with cage-mate. Also, rats tested with unfamiliar Longevans rats did much less prosocial behavior. The only difference in ultrasonic vocalization we saw in rats tested with unfamiliar vista rats, they did less 50 kilohertz calls that rats tested with cage-mate. The last question was if social buffering in our model can influence the extinction memory. As we know, extinction is a form of memory which is quite persistent. So to answer this question, we prepared the experiment where the rats, unextinguished subjects, were tested together with their partners, but then were retested next day and 1 week later alone. In the control group, the unextinguished subjects were tested 3 times alone. So here we have results. We observed strong inhibition of fear in rats tested with partners as previously, but this effect was not present the second day. when the rats were retested alone. When the rats were tested second time without a partner, they did much more freezing than the day before. So there was no social buffering, where the partner was not present. We also saw the analysis, we also did more detailed analysis of behavior, and we obtained very similar results. We saw some effect on prosocial behavior, some non-social exploratory behavior, but only on the first day. This effect was not present the second day. And the same was true for ultrasonic vocalization pattern. When the rats were tested with partner, they emitted mainly 50 kilohertz calls, but almost not 22 kilohertz calls. When they're tested the second day, they emitted the same amount of 22 and 50 kilohertz than like rats tested second times alone. And much less 50 kilohertz and much more 22 kilohertz than day before. So to sum up, the presence of a partner decreases fear response of unextinguished subjects, so the partner can buffer fear response of stressful animals. The rats tested together emit more 50 kilohertz positive ultrasonic vocalization and much less 22 kilohertz than rats tested alone. And the social inhibition of fear we observed cannot be explained only by changing the social context. For social buffering, crucial is the presence of a freely behaving partner. Anesthetized partner was not sufficient for this. Visual cues only partially mediate social buffering, and they are not crucial for that. And the unextinguished partner seems to be less effective in social buffering than extinguished one, but also naive one, unexperienced one. And familiarity and strange sameness influences to some extent social buffering in our model. And finally, the presence of a partner inhibits fear response, but this effect is not sufficient to improve fear memory, but because it's not lasting. And finally, I want to thank people involved in this project. First of all, my boss, Ewelina Knapska, Konrad Danielewski, Karolina Andraka, Ksenia Mejza, Kacper Kondrakiewicz, Joanna Sadowska, and Jan Kamiński for the Laboratory of Neurobiology of Mind. Thank you for your attention.
Any questions to Tomasz?
Yes. Yes. I did only for on males. There were one paper from Kiyokawa lab did on females, and they saw no differences in social buffering. So I use only one sex.
Because we have to ask it here because
yeah, thanks for a very nice talk. My question is social buffering. What would be the adaptive advantage of social buffering, if there is any?
So too much stress is maladaptive. So when you diminish your stress level by the presence of the partner, you improve your survival skills, your health, and so on. But social buffering is also good for established social bond in society and also in animals. So
stress would be stress in a dangerous situation. If there's too much social buffering, you might ignore danger that might be really dangerous.
A matter of balance between the stress and social buffering level.